to our distinguished guests, amazing viewers, and my dear colleagues. I am Yasha Nirati, currently working as a scientist in Biocon Limited, located in Bangalore, India. Speaking on behalf of Nyberman Bioinformatics, I am thrilled to welcome each one of you joining us today from different corners of the world for today's webinar on microbes in motion. Your presence here is a testament to our commitment to advancing knowledge and fostering collaboration within our scientific community. So today's webinar will help us explore how microbial communities respond to seawater flooding along with a natural salt marsh terrestrial pasture gradient and predict the microbial diversity using machine learning as a tool. So now I would request my co-host today, Ms. Lena, to take it ahead and help our viewers know more about Nyberman Bioinformatics and introduce our guest speaker as well. So Lena, please take it ahead.
Thank you, Ms. Yasha. Hello, everyone. I'm Lena Enya, a bioinformatician and a bioengineer. Welcome to Nyberman. We're not just a search firm, we are a global force connecting academia, industries, researchers, and students. Our mission is to break boundaries and let you explore diverse fields such as omics, health informatics, molecular modeling, programming, artificial intelligence, machine learning, and more. Over the years, our impactful workshops and 300 plus webinars have engaged more than 500 participants each, promoting collaborations and offering projects opportunities. I advise you to check out our website, nyberman.com, for more information. As we move ahead, I'd like to give a brief introduction to Reproducibility Lecture Series. As part of our services, we provide you with these lectures 2 times every month. If you're wondering, it's not just for Ph.D. candidates, it's for everyone, from bachelors to master's students to researchers diving into bioinformatics and its related fields. You can stay updated and be part of the series at nyberman.com/webinars. Before going any further, it's important to us to share some important instructions for you to get your certificate. First, you need to request the feedback link via Instagram or LinkedIn by sending us just one message, please. Second, the feedback link will be provided during the webinar that is now held from 11:30 a.m. to 12:30 p.m. And for Instagram message requests will accommodate until 5:30 p.m. And the feedback link will be active from 12:30 p.m. just after the webinar. As we dive deeper into this webinar, please meet our guest. Today, Dr. Sharon Rose Lopez. So, Dr. Sharon Rose Lopez is now with us. She is an accomplished life scientist with expertise in genomics and environmental sciences. She has a track record of leading innovative environmental projects and expertise in transforming complex data into actionable insights. Beyond her local impact, Sharon's international experience spans Europe, where she provided training and subject matter expertise, offering us today a unique perspective in today's discussion. Before handing it over to Dr. Sharon, I'd like to inform you, all the participants, to please collect the questions and keep them ready for the Q&A session that will be held later. Let's warmly welcome Dr. Sharon today for this webinar. Dr. Sharon, the floor is yours now.
Thank you very much. Hello, everyone. Thank you, Nyberman, for inviting me. And I'm really excited to talk about a little bit of my research, of one of my projects. And yeah, just I'll present you my work, and please let me know if you have any questions, and I'll try to answer as many as I can. Okay, so I'm going to share my screen now. Yeah, that's it. Okay, I think we are now there. Okay, fantastic. So now, as just read on the description, I'll be talking about microbes in motion and how machine learning is helping us to have different insights into the dynamic environmental changes. So for this presentation, I will try to keep it simple, but I have lots of information. So again, try to just to keep your answers at the end, and we'll try to answer as many as we can. So we will start. I will start with the importance of microbial communities. I will also describe why it's important and how we can use machine learning to discover more about microbial ecology. The methods I used for a project that I recently published, the results I got, and we will find some conclusions. So let's begin. One of the first things that we need to consider is that the climate change is affecting like many aspects of our lives, and of course, when we think about all the environment, we need to think about the coastal areas, like everything that surrounds the continents, and we can see that the climate change is affecting in so many ways. For example, first of all, we have the sea level rising. We have an increase of carbon dioxide. We have now more intense storms and also hurricanes. We have heat waves. So we have different ways on how the climate change is affecting, like all of these coastal areas specifically. For example, we also have seen like even more specific for specific coast areas. We can see that the sea level rises are accelerating the erosion of the salt marshes. When I'm not sure that the picture is too clear here, but we can see the effects of erosion, how the sea levels increasing are creating even more a higher rate of erosion in these areas. We also have an increased rainfall, which is increasing the sensitivity of till deposits and the stabilization of the areas. And also, we have floodings that can affect habitats and affect other plants and animals who are living in these parts. And just in case that we need to know, I will mention the word salt marsh very often. So just in case you're not familiarized with it, a salt marsh is the coastal wetlands that are flooded and drained by salt. It means that it's the areas that get where the sea rises, but then decreases. So that's what I meant when I say salt marsh. So now, why is it important to study microbial communities and the changes that are occurring here due to climate change? Oh, sorry. I'm so sorry. I don't know what's happening. Yeah, okay. Why is it important? Okay. Why is it important to think about like considering all the effects of climate change? We need to also consider. So sorry with this. Yeah, sorry. I have lots of tiny screens here. Okay, so why is it important to consider also the effects of climate change on microbial communities? Well, it is important because microorganisms play different important roles in the climate, in the environment. The microorganisms help in the composition of chemical components. They also help obviously in nutrient cycling, such as nitrogen, sulfur. So yeah, they're very important. They also help to regulate the salinity in the salt marshes, and also they have symbiotic relationships with plants and among other things. So that's why the role of microorganisms is very important when we think about the effects of climate change. And well, how do we do it? How can we control the effects of climate change? Well, so far, many organizations have tried to control the effects by reducing emissions from ocean-based transport, such as boats and like big ships. They also trying to conserve and restore coastal and marine ecosystems. And yeah, among other actions, they're trying to tackle the climate change. But we need to think not just in how to tackle the effects, but also how we can measure or monitor these effects. Right. So far, as we said, we also need to monitor, to control, we need to find a way to measure the effects of climate change and how to create a potential prevention. And we use different methods. We have satellites, we use observation in situ, we have models, we have different ways to control the effects of climate change. But well, and thanks to these methods, we have now lots of data. We have the little tons of data. But the question now is like, what can we do with all the data that we have collected? We can see, for instance, I don't know, like the sea level is increasing, but what does it mean? And we can have the measurements of how the sea has increased for in over 6 months, but what can we do with this data? And the answer to that is that we can use artificial intelligence and machine learning to analyze all this data. And artificial intelligence, we can define it as the simulation of human intelligence processes created by machines, especially by computer systems. And artificial intelligence is like a big component that has small subsections of the subdivisions, including machine learning and deep learning. How can we define machine learning? Well, machine learning is, as I say, is a subset of AI, or artificial intelligence, that uses advanced algorithms that can process all the information that we have and recognize patterns, and can help us to understand how the changes are occurring, and maybe even give some predictions. So now, how can AI and machine learning help us to understand microbial communities? Well, as I said, we have lots of data already from all the methods that we have used to monitor the changes on the environment. But what we can use is we can use tools from AI and machine learning to analyze all this data, to be able to extract the information and maybe create models or visualize all the information. And for machine learning, this is just a brief description, so don't worry. I think at some point in the seminar we will discuss this on more detail. But machine learning, we can classify it as supervised and unsupervised system parts, like models. And in very simple terms, machine learning supervised is when we have all the information with labels. That, let's say we have a table with columns, and every column has a name, so the model knows what it means. For example, sample 1, 2, 3, and we are controlling bacteria 1, 2, 3, 4, 5. So that's supervised. And then supervised is just we can provide the same data, but we don't have any labels. So the computer has to create a pattern without knowing exactly what is happening there, just to find what we can reveal by this information. Again, we will discuss this much later, but yeah, this is like in big detail. This is what machine learning supervised and unsupervised models are. So now, again, how can we use AI to understand microbial communities? For this, when we use machine learning and AI, what we need to do is basically, we follow this pipeline. When we have an idea, we then collect all the data from different sources, from different tools. We can identify the tools that we want to use, the models we want to use. We need to prepare our data, and then we can finally create a model and perform the evaluation of this model, and then we can just share our findings. And in more specific terms, specific terms for a project like this, they have been so many projects for people working with these topics, with environmental topics. And the process is, as I say, it's just begin with the sample, collecting samples, processing the samples. In this case, in the case of my experience, we analyze DNA or RNA. But I mean, obviously there are many other elements. We get the information from sequencing, and again, we have lots of data. We prepare the table. We prepare our data by cleaning it and processing it, and then we can have a model. So as I say, we can uncover patterns, we can identify trends, we can make predictions even. So that's how we can use machine learning. So now, as I say, well, this is more of the same. But with machine learning, we can identify patterns of relationships within the data sets. We can classify the samples. We can at some point predict the outcomes, and we also can create more data sets for data meaning at some point to uncover hidden patterns. But again, despite machine learning, it's increasing rapidly, and we have new technologies every day. We also have some limitations when we try to use machine learning for environmental monitoring. For example, we have one of the problems is that we have limited replication. Maybe we have problems with sample depth, and anyway, we have lots of problems. And most of the issues with machine learning is where the samples come from, like how we analyze the samples, because we will always our model will always be as good as our data is. So if I only can collect 10 samples, but instead of collecting 100, then we have some issues to create our models. So that's why we have some limitations with machine learning. But I think so far we are doing a great progress, trying to use the tools as best as we can. So now I want to talk to you for a specific case, for a specific research project that I led a couple of years ago. And part of this project has been published, not all of it, but yeah, most of this has been published. So if you want to have access to it, to the paper, just let me know. But yeah, so sorry. That's it. Okay, so in this project, we studied the effect of flooding events and how they can alter soil properties in a coastal area in Southport in the United Kingdom. And yeah, this is basically the area that we studied. It's called Southport. As I say, it's on the west side, nearby Liverpool. So yeah, for this project, we divided our samples in three main areas. We had the coast, and we look at this part of the picture. We had the coast, the salt marsh, that we call it the coastal area, and we divided it as a low area, a high, and after 20 meters away, there was an area where the people tend to where there's a pasture area. So we have these three areas for the study: low, high, and pasture. And what we did is we collected samples from these three areas, and we created a mesocosms. We literally have flasks where we exposed the soil samples from these three areas, and those samples were exposed to flooding, which is the seawater for a certain amount of time, for 0, 196 and 192 hours, and then we remove the water. We kind of remove the flooding, and we observe both patterns, the flooding and the recovery times, the recovery effects, and how we evaluated some chemical patterns, but also in my case all the microbial community patterns, to see if there were some changes, and how and my main goal was to evaluate the microbial community at the beginning, during the flooding and the recovery, to see if the microbial communities were able to recover, to maintain, to change, to recover, what was happening. And that's it. So again, and to do that, what I did is we analyzed the microbial composition by sequencing the 16S rRNA gene, and obviously with lots of informatics analysis. We evaluated enzymatic activity by measuring the concentration of cellulose, glucose, and sulfate, and we evaluated the resistance and resilience based on the microbial composition. As I say, we wanted to check if the microbial communities were able to maintain after the flooding and to recover when the flooding element was removed. So yeah, that's how we calculated the resistance and resilience. We used these two formulas, having published for resistance and for resilience. And the next part of the project was to create a machine learning model using a random forest to be able to predict how, if there were any factors from any chemical factors that were affecting more on the resistance and resilience properties. Because as we'll show you in a minute, that we observed that some communities were more able to resist or to recover after a certain time. So we wanted to see which factors were involved in this recovery process. So what did we find? First of all, when we evaluated the microbial composition and richness, for this step, we created a cluster model, an MDS graph. And what we wanted to see here in this part, we just wanted to see how the microbial communities were associated with, how were related. And again, this part, the red color represents the low area. Yeah, the low area. The green one represents the high, and the blue one represents the pasture, the other area. And so far, we can see that the microbial communities tend to be closer, tend to be like clustering the same with the same composition. So which means that the microbial communities in that sense are not changing much despite the flooded conditions, the flooding and recovery conditions. And then when we look at the microbial composition and richness, what we found is that, again, the microbial composition was slightly similar. Again, the red dots represent the low, the green ones represent high, and the blue ones represent pasture. And again, we can see the microbial composition, despite having different, like bigger abundances, relative abundances, we still can see that they have the same patterns, dominated by the same kind of, the same phylum, phylotypes. So yeah, in that sense, was kind of similar. And when we evaluated the enzymatic activity, again, with the only difference we noticed that was like relevant was that the pasture, the blue bars, representing a higher oxalase activity that it represents for that it's the way we measure cellulose. So that was the only difference, and the other enzymatic responses measured by this is glucose. The other responses were basically the same, so there was no more differences. So we wanted to see like, okay, so we have this information, and now we calculated the resistance and resilience. And again, for this graph, I know can be kind of confusing at some point, because we calculated the resistance and resilience with the graphs with the formulas I showed before. But yeah, anyway, so we discovered that the pasture area, where the blue bars had less resistance. We can see that at the beginning they are not, like, they're very low, even negative values, but at the end we have a higher resilience. And also we observed the high area, the green ones, it was able to recover as well, had low resistance at the beginning, resilience, but then it was able to recover. And the low area showed, but like it was like the most affected area, because all the times the bars were in like negative, showing negative values for resistance and resilience. So it's like the most affected area, the low, the one that is mostly exposed to the seawater. So maybe that's why it has this effect, basically by the water increasing for a long time and a longer exposure. So now, what we wanted to see, again, as I said, we observed that the three areas we studied had different patterns for resistance, for resilience. We had similar composition for microbial communities. We wanted to see which factors were actually affecting the resistance and resilience of these areas. We wanted to see maybe one of the enzymes, maybe the studied area. I wanted to see what was really happening that would help, that would create the best conditions for resistance and resilience. And in order to do that, what we did is we decided to use to create a machine learning model. We created a data set, we processed the data, and we ran a model, and we evaluated the model. And we wanted to see what do the results mean now? And for doing this, I selected a random forest model. A random forest model, it's a way where we combine an output of multiple decision trees. Again, I know this is maybe new information for most of you. We will discuss this in the following seminar. But yeah, it's a way of how we combine multiple decision trees and to reach a single result. And basically what we do is something like this. We have our data, we have subsets with different trees, and then we can find a consensus, a final result. So I created a Jupyter node, and I will show you the Jupyter node, just the model, just to show you like how it worked. Give me a minute. Yeah, it's here. Right. I guess everyone can see it now. Okay. So in order to create this model, in this case I use Colab, which is Google Colab, but it's a way to create Jupyter nodes. And the first, again, we work with data from this project from Southport, and the first step was to upload all the libraries we needed for this model. I uploaded my file via my OneDrive, and when I got information from the data set, we have this table. So basically, what this table is telling us is the different sources, as called pasture high and low, the name of the samples I assigned, and we have the two enzymes to detect, to quantify the presence of glucose and cellulose. We have the sulfate content, and these 5 columns in the middle represent the 5 main phyla detected on the samples, and the resistance or resilience. As you can see, the values are between 1 and -1. Well, most of them are very negative, but yeah, -1 to 1, representing the resistance or the resilience that they showcasted in the study. I also have the number of observed bacteria, which is trying to test the diversity of the number of observed species of bacteria. The Simpson index, which is a way to measure the diversity as well. And we also have the number of archaeal species detected here. For this study, I didn't find eukaryotic organisms, which was unexpected actually. But yeah, so we have archaea, and again the Simpson index for archaeal organisms. So basically, this is how the data look like. And well, again, I will describe this with further detail during the seminar. But basically, what I did is just check I cleaned the data by checking the null values, by replacing the null values. And then I finally created my model, my random forest model. In this case, I used the features that the two enzymes, the sulfate and the source, 4 columns, to just targeting the resistance and the resilience, of proteobacteria and how it affected. I created my model. I split the model between train and test. And then I created it and trained the model. And the first time I trained and created the model, I had a problem. Again, I will discuss with you during the seminar why I had this problem. But anyway, well, let's carry on. For this time, let's carry on with the model, and I explain to you later if you want to what happened here. So yeah, so we created the model. It's okay. Yeah. So we created the model. And we asked the model to make predictions. We then evaluated the model, and let me see if I can show you the results. Yeah, so yeah, basically, I had the results, and yeah, I'll show you. I'll be back to the presentation because I want to show you what I created with this model, if that's okay. Let me see, just to explain to you, because I have all the details here. But yeah, I created the features, everything. Just give me a minute. I can't see the way. Sorry. Give me a minute. So, okay. Sorry, connect one. Yeah. So as I said, I created the model, a machine learning model for a random forest to predict which factors were more involved in interfering in the resistance and resilience of microbial communities. And what I found is, when I created the first model, we have this model, and in the x-axis we can see the actual resistance of proteobacteria, like the data I provided, the data, and on the y-axis we can see the predicted by the model. As we can see, the R square value is very low, it's negative, which means that the model is not explaining much, that the model is it's not showing us exactly what we wanted to see. So basically, the model is saying that the information we used to create this model is maybe not the best. But also something interesting that we found in this it's okay. In this model is that when we created the feature importances plot, which is this graph, which basically tells us what is the order of importance for the features. As I say in the model, we use four main columns to create the model. Which was the carbon source as measured by glucopyranoside. We have the sulfate, we have pyranoside, that's a way to measure the cellulose, and obviously the source, if it was high, low, or pasture. And what we found is that the most important feature to determine the resistance in this case was glucopyranoside, which means that the source was actually not that relevant in determining the resistance and resilience of microbial communities. But again, this model wasn't the best one because we had a negative value for R squared. So what we in this case, we try to do that. We have a different approach, and instead of targeting the resistance and resilience of proteobacteria, I targeted the number of observed species of bacteria. And what we found in this case, the model is slightly better. We can see that the R squared now is 0.42. Obviously, it's not ideal because we are looking for a higher value than 90, but the 90%, so 0.9. But in this case, we have 0.42, which explains like the variability in the data. It's better explained than the previous model, but still it's not ideal. And we also created the plot for feature importances. And again, we found that that in this case, glycopyranosides or cellulose, it's the main factor determining the resistance and the presence of the diversity of, in this case, the diversity of microbial communities, as it was to determine the resilience of proteobacteria. And again, we can see that there's the source, which means, again, low, high of and pasture, the areas where the samples were taken from. It has the less importance in when we determine the richness or the resilience of bacteria, which means that it doesn't matter if the samples were taken from the lowest part, the middle, or the higher area, for the pasture area. What matters the most when calculating the richness and the resistance of bacteria, it's the resources they have, the carbon sources, the cellulose, and all the metabolic sources they have. So now, I mentioned briefly that when we create a random forest model, we combine lots of decision trees. A decision tree is like it's a tree. It's like a graph created for every feature that we have. So a random forest, it's a combination of all the decision trees that we have on our data. So for this specific model, I want to show you briefly what a random forest looks like for this project. And it's a lot of information, as you can see, and it begins with one of the enzymes and how it's selecting like one option or the second, and then we have another one too. So we have all of the sources, all of the nodes that and the branches that we have for this model. We can say it's a lot. You don't have to understand all of this now, don't worry. But yeah, I just wanted to show you like how complex the information can look, and how we and how we can use it. And as I say, in this case, both models were not the best ones, but definitely gave us an idea on an insights of what we're trying to find out, what we're trying to discover about these areas. And so yeah, in conclusion, we found out that again, that the low salt marshal mesocosms had the lowest degree of community and changes. And the pasture exhibited an increased change during the flooding. We also discovered that the microbial communities were able to adapt to seawater inundation, and they try to adapt and to survive to the different changes in sea level water. And we also found that the microbial resistance, as I say, microbial resistance, resilience, and diversity, in this case measured by alpha indices, depend mostly on the carbon or metabolic sources, not in the area of study. As I say, like what we saw with this machine learning model is that even if the microbial communities belong to the lowest part, the middle, or the pasture area, which is slightly farther away, what matters the most for the microbial resistance and for the diversity is the carbon source availability, the resources they have to survive, and not exactly the area of study, which means that the impact they could have from climate change floodings can be like a handle and tackled by the resources they have. So it's, in this case, it's more important to think about the quality of the soils than how the sea is affecting and is impacting the communities. And also, as we can see, we discovered all this information thanks to machine learning models, because the information we have from the graphs we have from the experiments we ran was just that, okay, we saw the resistance and resilience changing over time. That's it. but it was thanks to machine learning that we were able to have new conclusions and to have new insights into what is actually happening. So yeah, machine learning was valuable and definitely can be a powerful tool. And we can even improve our models, because the models we have are maybe not the best ones yet, but we can try. There's literally a world to explore. So yeah, we can try different models and we can find better resources. So yeah, as I said, with the information we have, we can now we can have new opportunities. We can create innovative research for environmental monitoring, and we can get new breakthrough insights. So yeah, so from my experience, from what I see in this project and from what I've seen with other projects, I think the best way to study the effects of climate change and how it's affecting the quality of the soil, it's definitely by combining and integrating tools from machine learning or artificial intelligence with traditional methods. I think that's the best way to do to unravel the complexity of environmental dynamics. So definitely that's the best one. And so yeah, that's everything for me. So please, if you have any questions, we'll try to answer as many as we can. But if not, you can find me on LinkedIn, on GitHub, where I share the Jupyter node for this model, and also you can reach me on via email. Okay.
Thank you so much, Sharon. It was a lovely presentation, I must say that. Your presentation was truly exceptional, seamlessly integrating diagrams and photos to create an engaging visual experience. And your thorough exploration through the concept, delving into the methods and results within that singular case study was remarkable. So I personally found your expertise and achievements both impressive and captivating. And also the significance and relevance of machine learning in today's landscape is undeniable, and your session has provided our viewers with a fresh perspective and deeper insights into this vital and trending topic. So thank you for your invaluable contribution in expanding our understanding for machine learning. Thank you so much. And I'm very sure that the viewers or our audience must have gained substantial knowledge. So they might be having questions in their mind. So I would now request our audience to please post their questions in the comment section or the chat box. We'll address them one after the other. Okay. So there is this question from Monica. She is asking, is it possible to predict the microbial diversity in plant using the same artificial intelligence or machine learning at specific location? Like we only talked about the microbes, so she's asking for whether we can do the same for the plants as well.
Yes, in this case we can. And for this specific project, I was obviously focusing on microbes, on microbial organisms. But yeah, if we have the information of the diversity of plants, we can try to predict the presence or the abundance. Again, this can be it's possible, but definitely we need to we need to collect the data, but it's possible. And also it depends in how, like, what do you mean by predicting? Like, if you just want to see like how a model can predict at this point, but if you want to predict, let's say in the future, then we're talking about a different kind of approach for artificial intelligence, which is the deep learning. But that's a more complicated stuff. But yeah, it is possible to do it as well.
All right. So the next question was, is it possible to do the enzyme estimation studies of different microorganisms at a time?
Yes, it is. It is possible. In this case, I just measure the enzymatic abundances like in the environment. But yeah, definitely it is possible. If we track for example RNA, if we sequence RNA instead of DNA, we can track for specific enzymes or metabolic activities occurring within the organisms. So yeah, it is possible. In this project, I can try to do it. I can try to to find like specific patterns. But since it was just DNA, I don't think information will be very reliable, but definitely it's possible.
Okay. Okay. So for that project that you were talking about during your presentation, for that particular project, people want to know what all programming languages and bioinformatics tools you used.
Okay, for this project, in terms of informatics, as I say, I use sequencing of the 16S RNA gene, sorry. And with all the information I have, I handle that information on Kime. I'm sorry, I know everyone
Yeah,
calls
it's
it
kind.
different.
Yeah.
Kime, yeah. We all call it in different ways, but yeah, I use Kime to do all the analysis to prepare the sequences and to find the taxonomy. I use that tool. I also use PyCross, but again, as I said, because PyCross is a way to find the metabolic patterns, but as I say, it's DNA, so I don't feel very confident of that information. And after that, when I had the tables from Kime, I did the analysis and visualization on R using Philosec. So yeah, that's basically those are the main tools that I use to create this information. And for machine learning, as I say, I use machine learning on Jupyter Notes.
Okay, so before moving ahead, dear viewers, we also had a webinar on Kime. So in case you want to know about it in detail, you can go and look for it on YouTube and also on our website. Next was that there are many people who do not have the basic idea and they have never learned this about the programming and the machine learning concepts. They are at the very beginning stage. So what would you suggest the foundation skills one must have?
For machine learning, I would say that you need to have like a strong understanding of mathematics. Maybe not advanced mathematics, but you have to understand like some different like even statistics. When we talk about like how we can classify information, what we mean by clusterings, for example. Yeah, I think the basics would be like just some mathematics, and then I would say try to learn some coding. You can start with R if any of you are working on academia or doing a PhD or a degree. I think R is a good place to start. And then, yes, you will have to try some Python. You need to get like involved with Python to understand what is happening, what is it doing, and then we can start switching. But yeah, I think it's again, I don't have much experience. I have been working with machine learning just for a year. Sorry, so I can say that yeah, it's something that we can achieve. But yeah, we just need to work hard. But yeah, we all can do it.
Okay, but the only thing I feel is it requires a lot of practice. This is what I know.
Absolutely. And this model that I created, I have a couple of mistakes. As I say, I've been doing this for a year and still I have some errors. So yeah, and if I can share my GitHub with this model, I'll show you that I have a couple of errors. Like, oh, come on, simple errors. But you need to keep practicing.
But I think this is the way we all of us learn. I think that's the only way.
Yes. By error and mistake, yes.
Okay, so this next question was this person, Mohammed Umar, is working on Arctic microbial diversity, basically focusing on curable based and testing for several enzymes. So is it possible to do the same as you did? As in, maybe this question needs to be asked more appropriately. If you could explain a bit more, Umar, it would be better. We could not get your question correctly.
See, it's about like if we can create the same or similar model for the samples. Because if that's the question, I think it is possible. Actually, I'm currently working on a collaboration with a group who's working with Arctic samples. And yes, we are trying to achieve the same approach, creating a machine learning model to predict the composition.
All right. So the next question is, as we know, there are not many models. So for a random forest model, how did you choose one particular? As in, what are the parameters that you look for when you're choosing a model?
The main thing to consider when you're choosing a model is what you want to see, what you want to find. Like, first of all, which kind of data you have, which data, if you can quantify the data, if it's values, if it's words, depending on what you have for the data. In Python, we call it with different names, but so far let's call it words or numbers. And then what are you trying to prove with your model? Are you trying to just to visualize something? Are you trying to predict and to create, like, I don't know, like a regression model? What's the main algo? What do you want to prove with this model? So I think after that you can decide, okay, maybe this is the best option for me, or maybe try this one. So yeah, it depends on what you're trying to find, and also what the data. What is the data telling you? Is it data that it's different? In this case, I chose a random forest because I had data showing different values, showing resistance, showing abundance of enzymes, showing number of species. So it was different data evaluating different values. That's why I created a random forest that combines decision trees created from different inputs.
All right. Okay. So if anybody wants, they can unmute themselves and ask a few more questions. We can take some more questions. All right. We'll move ahead. I don't think there are any further questions. Okay. So thank you so much for addressing the questions, Dr. Sharon. So before you go, take a moment to hear about our upcoming internship opportunity. So join us for a 15-day virtual internship focusing on the application of machine learning in bioinformatics. And for further information, you can stay connected through our WhatsApp channel. You can join the channel through the provided link on the slide. So before we conclude today's enlightening session, I want to extend our sincerest gratitude to our guest, Dr. Sharon Lopez. Your invaluable insights, expertise, and passion have truly enriched our discussion today. On behalf of everyone here, I want to express our heartfelt appreciation for your invaluable contribution to today's discussion. Your wisdom has left an indelible mark in our minds, and we are truly honored to have you with us today. So thank you so much for coming, and we hope to have more collaborations with you in future webinars. And dear audience, you will soon receive a feedback link from our Instagram team. Please ensure that you provide your correct name and email ID when you complete the feedback form. Additionally, we kindly request all of you to share your certificates on your social media platforms, tag us, and help us spread our word about our upcoming events. So before leaving, I want to once again express my gratitude to each and every one of you here. We look forward for active participation in our future webinars. Until next time, bye-bye. Take care.
Thank you, everyone.
Thank you so much. Thanks a lot.